Shubhodaya. Many of you don't know me. I'm a new faculty at Department of Biology. Um, but today I'm not speaking, right? So I'm here to introduce Dr. Regina Baucom. So we are really excited and thrilled to have her today here. Dr. Regina Baucom is a professor of ecology and evolutionary biology at the University of Michigan. Dr. Baucom's research spans topics such as reproductive strategies that enable weeds to maintain fitness under strong selection pressure. I noticed one news that was based on her paper. The title of news is "Different Ways to Have Sex Yet Still Be a Weed." And also floral trait evolution in response to climate change, where plants increase investment in floral size and rewards to sustain pollinator interactions. And that was another news headlines. Bigger flowers, greater rewards. Plants adapt to climate disruptions to lure pollinators. I believe she will talk about some of these today. So Dr. Baucom has also demonstrated how herbicide drift, including dicamba, can disrupt plant pollinator networks. We heard about that yesterday. And her current research investigates the genetic basis of herbicide resistance in morning glories across diverse populations. You can see a beautiful flower of morning glory when we have this evening glory right here. Dr. Baucom, I saw somewhere, maybe she will talk about this today, also was involved in the 1,000 plant project. Looks like pretty big project that generated transcriptome data for over 1,100 species. Please join me in welcoming Dr. Regina Baucom today.
Hey, thank you for that nice introduction. I can't take responsibility for any of those titles that were developed by the news media, or I won't take responsibility for them. So can I get a show of hands? How many folks in here are grad students? Okay, awesome, because I kind of wrote this talk for grad students, so I have the right audience. Today I'm going to talk about lessons from weeds, how to resist and persist in times of stress. So I'm going to talk about what makes a weed, who are weeds, who are our weeds, and where did weeds come from, where did they originate. And I'm going to talk about what we've learned about evolutionary biology using weedy plant species. And then I'm going to kind of take a step back and talk about lessons that we can take from weeds and incorporate into our personal lives at this particular point in time. And so at one point in this talk, I'm going to ask you to step into the cringe with me, and you'll know when I get there. And we're just all going to be friends and step into the cringe together. Okay. All right. So what I wanted to do is not advance so quickly. What I wanted to do is first ask, when I say the word weed, what is the first thing that pops into your mind? Undesirable. What was it in the back? Did you say marijuana? That was what I was going for, actually. And then I was going to say, okay, what is the other next weed that you often think of? Like specific weed. Dandelion. Okay, right. Dandelion. So we are we're all familiar with dandelions. I was a little bit worried that dandelion was more of an Eastern weed, but I was happy to see some out here today. Dandelions are an interesting weed in that they've really driven an important part of the herbicide economy. A lot of herbicide development has focused on this particular plant species, trying to get rid of it existing in our suburban lawns. Nobody wants a lawn with dandelions, although I don't personally mind it. We also have a lot of other suburban weeds, such as field bindweed. So this is Convolvulus arvensis, false bindweed. We've got plantago, clover, and my nemesis in my garden, Creeping Charlie. Okay, so these are common weed species that we find in suburban lawn sort of areas, and you've probably seen them, especially if you spent time out east. We also have weeds, not just in suburban lawns, but in agriculture. And the other portion of the herbicide industry has really been fed by noxious plants, noxious weeds of agriculture. So up here, I'm showing you the common morning glory, which is the species that I most often work on, infesting a soybean field. And we can have really bad infestations of this species that make it so the farmer can't get in and harvest their seed in time, and it can lead to reductions in yield. I'm also showing you Rumex up here, and then a couple different amaranthus species. So these are some of the top 10 worst weeds of agriculture. The Weed Science Society of America every year asks weed scientists and farmers, what are the weeds that are giving you the most problems? Like, what's the weeds that you hate the most? And we have a top 10 worst weeds of agriculture and row crop agriculture. And I noticed something interesting. I mean, it's typically the same species every year, Amaranthus, Morning Glory. I think Rumex makes the list. I noticed that the species that are the top 10 weeds of agriculture, and I'm showing you some here, are also found in urban and suburban landscapes. So you can also find these species existing in, cracks in the concrete, right? And in just sort of waste spaces in areas of human disturbance. So I've become really interested in thinking about, I've always been interested in thinking about the traits that make a weed, but now I'm trying to think through what is it that makes these particular species capable of adapting to both agricultural spaces, suburban lawns, and then also urban areas, because that's a pretty different ecology for these species to all predominate. Okay. So today, the structure of this talk, I'm going to first talk about what is a weed. Why do we name something a weed? What have we learned about evolution from weeds? Which traits allow weeds to persist? And then what lessons can we learn from these weeds? Okay, so first, what's a weed? So Harlan and DeWitt published in 1965 a bunch of different definitions from the literature that ranged from the early 1900s to the 1960s. And they grouped them first by A. If you can't read that, it says, here are some definitions by professional weed men. And this second entry, B, by enthusiastic amateurs. And then C, by the ecologically minded. Okay, so a lot of people have different definitions of weeds. The most typical one that you see is a plant growing out of place. So that's the one that you kind of see consistently across the literature, regardless of if it's an ecologist or if it's a weed scientist. This is a plant that is growing in a place I do not want it to be. We also, a more recent definition is the plants that compete with crops for resources. You'll often see in these definitions terms that are negative. These plants are obnoxious, they're nuisances, they're unwanted, right? So there's this like designation by humans that we do not like these things. They are annoying. And then my favorite definition I've found so far, any plant that interferes with man's plan of conquering the earth. That's straight out of the 1950s, right? We're going to conquer the earth. The enthusiastic amateurs had a kinder view of weeds. So Ralph Waldo Emerson said a weed is a plant whose virtues have not yet been discovered. Okay, so even though our definitions of weeds tend to be like, we don't like these plants, they're nuisances, they get in our way, there's also a theme of, well, these plants, we like them. Like we just haven't discovered what they're here for, right? So there's also kind of a positive view. In current day, you still see people in just current culture using the term weed in different ways and in both variable ways. Okay, so outside of science, people are using the term weed to mean something. This guy recently, I don't know if I need to introduce him to you, right? So this is Elon Musk. He said, I think we do need to delete entire federal agencies as opposed to leave part of them behind. It's kind of like leaving a weed, he said. If you don't remove the roots of the weed, then it's easy for the weed to grow back. Okay, so this kind of encapsulates what we as scientists are dealing with right now, right? Where we have a group of people that want to get rid of the federal agencies that fund our research, right? That as scientists make our work possible. And he's likening them, federal agencies, to weeds. We got to rip them up by the roots. There's also a cultural perspective of weeds that is much more positive and one of like strength and persistence. So the Danish artist Bertha Wegman in 1910 painted portraits mostly of dandelions, right? And she painted dandelions as because she appreciated their uncontrolled, strong-willed growth and painted them as a symbol of women's liberation in Switzerland. Okay, and so she was a particular painter that was a cut above a lot of other painters. The men painters of the time they recognized her brilliance, but they definitely kept her as separate from them. One of the things that made her so different was because a lot of the painters of the time, if they're going for floral portraits, they're painting roses, right? And she decided to paint dandelions because of their strong-willed growth and their persistence, right? Okay, so back to the scientific definition of a weed. So, the term weed is in it has cultural relevance in different ways. But the scientific definition, the generally accepted one, is a plant growing out of place. So if you think about that, it's really the interaction or the overlap between how we as humans have an opinion about a particular plant species and its ecological context. So we do not like this plant in this place here. Right? So it's an opinion and an ecology. After we've developed kind of this definition, Baker came along, Herbert Baker came along and wanted to understand what were the traits that made a weed, okay? And so he took a bunch of different species from the Asteraceae and said that were weeds and said, what sort of traits do they have in common? And so he came up with this ideal weed trait list. And so the ideal weed characteristics are germination requirements are fulfilled in many environments. It could germinate wherever. Discontinuous germination, rapid growth through vegetative phase to flowering, continuous seed production, self-compatible, but can also outcross, high seed output. Okay, so he came up with traits that he thought were associated with being a weed. And he also developed this concept of the general purpose genotype, which basically means a genotype with a broad environmental tolerance and plasticity. So weeds had these ideal Baker's traits and also were tolerant and plastic of most environments. Okay, so that's for a while, or pretty long time, that's how we thought of the traits that differentiated weeds from non-weeds. Since that time, researchers such as myself have taken off and asked similar questions as Baker, but instead of just using members of the Asteraceae, we've done database searches, right? So a previous postdoc in my lab, Adam Kuster, we had a database of plants that weed scientists said were weeds. So we had this designation from the Weed Science Society of America, about 2,500 species. And we also had a database of all the US species of plants. And we had a database of traits from the USDA associated with particular plant species. And from that, we did some comparisons between weeds and non-weeds to assess across all the species, not just members of the Asteraceae, what are the traits that lead to a plant being a weed. And what Adam found was weeds typically are annual, they have fast growth, high fruit abundance, fruits that persist on the plant, rapid seed spread, high seedling vigor, fast vegetative spread, high calcium carbonate tolerance, and high salt tolerance. Now, a lot of you in here are probably going like, okay, well, she's talking about weeds. What about invasives? We also did that too, but let me explain what we did. So we also knew the introduction status of these weeds in the database. Okay, and so we were able to contrast introduced weeds to introduced non-weeds. and native weeds to native non-weeds. So the traits that I'm showing you here for introduced weeds are from the contrast of introduced weeds to introduced non-weeds. In the introduced weeds are what you would think of as invasives, okay? So our introduced weeds have high salt tolerance, fruits that disperse from the plant, and they're more likely hermaphroditic. And so hermaphroditic basically means that a plant flower has both the male and female parts, and it can produce seed by itself, okay? And so this is kind of a combination of Adam's work and also another postdoc from the lab, Megan Van Etten, who did this database survey to look at different breeding systems associated with weediness. In terms of native weeds, we found that native weeds exhibited low shade tolerance. They showed high calcium carbonate tolerance, fruits that persist on the plant. Okay, so in comparison to introduced weeds, native weeds had fruits that persist on the plant. I thought that was kind of interesting. And they're more likely monoecious. And so monoecious basically means that a plant has flowers that have the male parts and then flowers that have the female parts. Okay, so weedy plants, whether you're introduced or you're native, you're going to have the equipment that you need to produce seed in a new environment. Okay. All right, so those are traits or weeds. So who are the weeds, right, in our databases? Well, we found that every plant family has a weedy plant member in it. So weeds are across all plant families, but there are particular genera that are particularly enriched with weedy plants. And so these are the 15 plant genera that have a higher proportion than you would expect of weedy plants. I don't know if you see any genera that you particularly like up here. You've got some morning glories, got some chenopodium. Anyone see anything that they find they like? Plant genus? No one in the audience? Like, no. Morning glory. Okay, well, Ibomea, right? Got solanum, centurea. That's a big weed out west, I believe, right? Yeah. Okay. Amaranthus is a big one. And acacia, that's a big one. Brassica, I mean, there's so many brassica species, right? Okay, so 15 genera are enriched for weedy plants. Where do weeds come from? Okay, a really nice review article from Anna Caicedo's lab, along with Ken Olson and a postdoc at the time, pulled together some ideas of how did weeds evolve, right? And they showed three major pathways for weeds to originate. One was that, you just have wild species. They become accustomed to human environments. Through that, they become weedy. The arrow that I'm showing you is supposed to give you an idea of the amount of genetic variation within the species. So the fact that this arrow is big means that the transition from going from a wild species to a weed, you probably retain a lot of genetic variation. And the majority of our weeds probably came from this pathway. Another pathway is the crops that we plant. So we plant some crops out. There can be mutations in the genomes of those crops, and they can de-domesticate to become weeds. In this situation, you would predict there would be low genetic variation in that weedy species. And the most common situation that we've seen is weedy rice, where we have de-domesticated rice that are weedy in normal rice fields. Another pathway is wild species can cross with crops and hybridize to produce a weed. The most common example of this is wild and weedy beets. Another example that I think we need to think about a little bit more in depth is foraged or cultivated species. So in medieval times, people were basically foraging dandelions, lamb's quarters, field bindweeds, amaranths, all species that are currently the top 10 weeds of agriculture now. So they were foraging them in medieval times for vitamins. Basically, these are some of the first species that would pop up in the spring, and they'd take the leaves, for example, of dandelion and make a tea out of them to get vitamin A. Chenopodium, lamb's quarters and amaranths had a really high protein content. Okay, so these species that we consider really awful, noxious crop weeds have been in association with humans since medieval times. And even we've got records from 1800s where Emily Dickinson is cultivating dandelions in her garden, like on purpose, right? So it was used even then. Okay, I'm wondering if we shouldn't change the definition from just a plant that's in a place where we don't want it to thinking about it in terms of weeds as human commensals, okay? So Fosserup et al. Had this nice tree article that come out this year where they defined human commensal species. So a wild organism is a human commensal if it's a species that depends on anthropogenic resources, like disturbed areas or food, you know. Another part of their definition is that they require that there has to be genetic differentiation from the ancestral non-commensal form, which I think is kind of a high bar. But to give you more of an in-depth of what they're thinking, so species that are non-associated with humans might be like deep sea fish. Okay, species that are synanthropic use human resources opportunistically, right? They tolerate humans, so their concept of that is like the raccoon or blackbird. Human commensals really depend on anthropogenic resources and they're adapted to those anthropogenic resources. So I think, you know, weeds in agriculture that rely on disturbance regimes to be able to have a, you know, normal life cycle are probably human commensals, right? You could probably argue that other weeds might be more synanthropic or just opportunistic, right? Okay, so if I was going to redefine weedy plants as human commensals, I would have it formatted this way, where I would say weeds are plants that are able to take advantage of the environments that humans create and manipulate. An important component of this is that the species can plastically or through adaptation reproduce in the human disturbed environment. Okay. Okay, so that's what weeds are, who our weeds are, where they come from, a few different paths that have been suggested. Next, I want to talk about what we've learned about weeds or what we've learned about evolution from weeds. And of course, we're going to start with Darwin. Okay, so Darwin studied common morning glory. It's like so excited about this, right? If you are an evolutionary biologist and Darwin studied your species, you're like, oh, it's special, right? No one's nodding. So you're like, I don't understand what you're saying. Okay, so what he did, he studied inbreeding depression using the common morning glory because it's hermaphrodite. It can both self and outcross. He did 10 generations of outcrossing and selfing. And what I'm showing you here are you know, a picture of plant heights. The plants that were outcrossed were much taller. The plants that were selfed are much shorter. Okay, so that's just evidence of inbreeding depression. He also did this thing called the weed garden experiment where, you know, he literally made just like a, I don't know, space in his lawn and then looked to see what germinated and watched, I can't remember if it was like 200 or 600 germinants, but he watched them over time to see who survived and who didn't. And of this initial group that started, you know, you had some taken out by snails, et cetera. And so it was supposed to be the first example that he was watching natural selection with a group of a weed community. He also used weeds, so this is Oxalic stricta, to study this concept called leaf sleep, which is, you know, every time I think about this, it just kind of blows my mind. A normal leaf of Oxalis looks like this, where the leaves are open to the sun. And leaves that are sleeping, the leaves are pulling back and they're going vertical, not horizontal. And so the leaves actually move. He hypothesized it was due to changes in radiation and to keep the leaves from being exposed to changing temperatures at night and frost, right? And since this time, we've actually uncovered the genetic basis of the ability for these leaves to move at night, right? Okay, so that's what Darwin did. Recently, I searched the literature for, I went to the evolutionary biology literature, like molecular ecology, evolution letters, PNAS. I think I included another one. And I searched the terms weedy plant, weed, and then some specific species that I knew were weeds that people have been working on for a few years. And what I wanted to do was to understand what kind of questions in evolution we're asking using weedy plant species. I didn't include weed science journals or weed research journals because I wasn't trying to look at sort of the more management angle, just the evolutionary biology angle. And so I pulled all this together, and over a space from 1960 to the 2020s, we've got about 200 different papers I was looking at. And in the 1960s, we started just sort of foundationally describing systems and doing some descriptive cytological work. In the 1980s, we start talking about an evolutionary and invasive lens, asking questions like, do polyploidy and hybridization explain invasiveness? In the 1990s, we moved to really focusing on mating systems and gene flow. So asking, how do mating systems and gene flow shape adaptation? In the 2000s, we took a molecular turn and started asking explicit questions about plant adaptation, such as what is the genetic basis of weed success? In the 2010s, we started working with an integrated eco-evo framing, and also an applied framing started to make an appearance in the evolutionary biology literature. And I think that's because people that were working on the more applied angle started working more in evolution and then publishing in the evolutionary biology literature. And now in the 2020s, we're asking questions like, can we forecast adaptation and manage it evolutionarily? And we're taking a multi-stressor, genomic, and as well management heavy approach at this point, okay? The major research topics between this time, the majority of the work, so this is just the percentage of records by topic, the majority of research was focused on mating system, gene flow, and hybridization. So we were doing a lot of work just on mating system evolution using weedy plants. Biotic interactions was also a big category, genetic and genomic architecture of adaptation, and still kind of a lower level, evolutionary management and applied evolution, rates and repeatability of adaptation, multi-stressor and global change interaction, as well as some questions about plasticity versus genetic change. Okay. If you want to look at what major themes were across these papers in terms of the questions that people were asking, 4 major themes popped out. 1 is evolution is rapid and repeatable. 2, evolution is shaped by constraints. So there's a lot of constraint work done on weeds. The 3rd is plasticity and polyploidy are important for persistence. And 4 is human altered landscapes are evolutionary laboratories. So I'm going to give you a couple of examples of some work from my lab that fall into these, at least 3 of these major themes. Okay, so in terms of evolution is rapid and repeatable, I think one of the best examples of rapid evolution and repeatable evolution comes from pesticide resistance evolution. And the evolutionary biology literature was influenced heavily by insecticide resistance in insects. In 1937, Dobzhansky said that cyanide resistance in the red scale was the best proof of the effectiveness of natural selection yet obtained. So that's like a pretty big deal that, pesticide resistance was considered the best evidence of natural selection by Dobzhansky. Then about the 1950s, Jim Crow and colleagues started studying DDT resistance. And they were asking questions about genetic adaptation or genetic basis of DDT resistance in fruit flies. And about this time in 1956, the ecologist J.L. Harper predicts the evolution of herbicide resistance. So we kind of had a start in the evolutionary field thinking about insecticide or pesticide resistance. And then in the 1950s, J.L. Harper said, we're probably going to have weeds that evolve herbicide resistance. We did have our first example of herbicide resistance evolution in 1957 in sugarcane plantations in Hawaii. And then in 1970, triazine resistance really started to take hold first in Convolvulus arvensis, so that's field bindweed, and Senecio vulgaris. And then we can see, so this is the number of unique cases of herbicide resistance over time, chronological increase of resistant weeds globally. And so you can see that from the 1970s onward, we just kind of have an exponential increase in herbicide resistance, unique cases of herbicide resistance over time. Okay, so evolution is definitely repeatable and rapid. And some of my own work has hit on this theme, where with the common morning glory I showed yesterday, we have multiple populations of the common morning glory that show high levels of resistance to Roundup or the herbicide glyphosate. So that means it's repeatable, right? So it's found across multiple separate populations. It's also, we found that it's rapid. So again, Adam Kuster, in a previous publication from the lab, he was actually, when he did this survey over here, he actually had not just a spatial sample of populations, but also a temporal sample. So when I was a grad student, I collected these populations in 2003, and I held on to those lineages. He went back to the populations and recollected from those locations in 2012. And so we did a dose response curve, looking spatially as well as temporally. And what I want to point you to is that in a span of 9 years, this area in the southeast, in the coastal plain of North Carolina and South Carolina, where it used to be in 2003, pretty susceptible, all the populations had pretty low resistance. We started to have high resistance in those populations after 9 years. Okay, so evolution is rapid and repeatable. We've tried to uncover the genetic basis of resistance. We've done surveys of the genome. We've found areas of the genome that show strong signs of selection, and within those regions, we find detoxification or herbicide detoxification genes that are showing strong signs of selection. Here on chromosome 10, we find a haplotype that is shared across our 4 resistant populations that we sequenced. So we think that we have evidence for a haplotype that is the same across resistant populations. So it's repeatable, but also the genetic basis is probably shared. Whereas other haplotypes that were showing strong signs of selection that also had evidence of detox genes within those regions were not shared across populations. Okay, so we have some evidence that the genes underlying resistance in this species are shared in some sense, but because this is a polygenic resistance mechanism, there are other haplotypes that are not shared between the resistant populations. Okay, and that's work from Sonal Gupta as well as Megan Van Etten from the lab. If you're interested in repeated evolution, especially in terms of herbicide resistance evolution, I summarized the repeatability of herbicide resistance in a paper in American Journal of Botany. So if you want to look up some good cases that's beyond morning glories, I would look there. The second topic is evolution is shaped by constraint. And so what are constraints? Constraints are effectively brakes on on evolution of any particular trait. In this case of herbicide resistance, we're looking for, are there fitness costs associated with being resistant to herbicides? So that would put a brake on the continued evolution of resistance. With morning glory, with the populations that were high resistance. So I'm showing you on the x-axis survival at a high dose of Roundup. Populations that were the most resistant over here also showed a really low proportion germination. So we believe we found a fitness cost in the common morning glory associated with glyphosate resistance or Roundup resistance, but it incurs a fitness cost, which means that if you don't apply the herbicide, those lineages that are herbicide resistant should be at a disadvantage compared to susceptible types. And that's part of what people put into rotations when they're trying to manage the evolution of herbicide or any kind of pesticide resistance. I'm just going to skip over that. A really important paper came out in the late 1990s by Joy Bergelson and Colin Purrington, and they were trying to assess constraints in terms of fitness costs associated with resistance in plants. And what they found is that for any adaptation, whether it's herbivory resistance or herbicide resistance, only about 50% of the time are researchers uncovering fitness cost. And that kind of blew people's minds. And people working on insecticide resistance in insects have found the same thing. We don't always find fitness costs associated with a particular resistance allele, which is bad news for control and also kind of confusing, right? So why wouldn't we uncover fitness costs associated with an adaptation? Typically, we're thinking it's like a new adaptation that should be selected out. And a summary of the literature from my colleague Paul Neve. He looked across a bunch of different systems and said that fitness costs were really contingent, at least in herbicide resistance examples, on the resistance mechanism, the specific resistance allele, the dominance of the resistance cost, the pleiotropic effects of the herbicide-resistant target proteins, the genetic background, and the environment. So there are a lot of different things that can vary for fitness costs to be apparent, okay? And so that's a lot to learn about the nature of fitness costs, right? Just from the herbicide resistance literature. Okay, and so the third topic that was covered by my database survey was human altered landscapes are evolutionary laboratories, right? So basically that's a fun way of saying you can, in human altered or human mediated selective regimes, we can have all sorts of consequential types of evolution that we wouldn't expect, right? And so a good example of this comes from work from Nia Johnson, one of the previous students or PhD students from my lab. And she was working with velvet leaf, which is the species that she's, I guess, counting insects on over there. It's velvet leaf, Abutilon theophrasti. It is interesting. It's called velvet leaf because its leaves are really velvety, and they're really velvety because they have 4 different types of trichomes. Okay, and so high density of trichomes and 4 different types of trichomes. And so the different types of trichomes are, you can see up there, the C indicates branched trichomes. We've got single trichomes that are just like a single trichome. We've got peltate trichomes and capitate trichomes. So 4 different types of trichomes. And what she was interested in is to understand, are these trichomes serving as an herbivory resistance trait? Are they mediating, keeping basically the insects away from the leaf so they're not able to herbivorize the leaves as much? And so over here, she's showing that as you have a higher proportion of branched trichomes on your leaves, you have a higher level of herbivory resistance. So that shows there's a correlation between those two things, suggesting that these branched trichomes function as an herbivory resistance trait. She also found a positive, it's a slightly lower correlation, but still a positive correlation between the proportion of branched trichomes the leaf had and its level of herbicide resistance. Okay, so the herbicide she was using was glyphosate, so Roundup. And so this suggests that branched trichomes could be functioning as an herbicide resistance trait. We think possibly if it is functioning in this way, we've got these branched trichomes, perhaps they're keeping the herbicide away from the actual leaf itself. And she also did manipulative fitness study, where she had plants growing in the presence and absence of the herbicide. And she found positive selection in the presence of herbicide on the proportion of branched trichomes on a leaf, right? So plants that had more branched trichomes had higher fitness in the presence of herbicide. In the absence of herbicide, this relationship changed. So if you had higher proportion branched trichomes in the absence of herbicide, you had lower fitness. So the fact that we changed this environment, and the environment is the presence of herbicide or not, the relationship between fitness and the trait changes. So that means that herbicide can actually act as a selective agent on the proportion of branched trichomes. Okay, so that was kind of a mind-blowing finding, or interesting finding. Along with this theme, another student from the lab, Grace Zhang, she's now a 6-year student in the lab. She's working on the intersection of herbicide and herbivory resistance in the common morning glory. And one of her first chapter, we showed that plants that were treated with glyphosate of the common morning glory show a much higher proportion of damage by herbivores. So you hit them with herbicide, plants don't die, the herbivores come in and like to eat them more. We thought this was really interesting. It could be changing all kinds of ecological stuff that you ecologists would know about. And she also looked at the type of damage in terms of, is the insect creating holes in the leaf? Are they, eating the margins of the leaves, or are they just eating the surface of the leaf? And what she found is the pattern of damage by, potentially different herbivores changes in the presence of herbicide. So in the presence of herbicide, you have less surface herbivory, but you have more whole herbivory and more margin herbivory. So we're wondering if not only does the herbicide make the plants more tasty or something to the herbivores, but it's also changing which herbivores are willing to eat on the plant. Okay. And getting to the end of the talk, I wanted to cover which traits allow weeds to persist. So a lot of the work that I've done, not just working on traits associated with weediness, but also traits associated with plant defense, right? So I wanted to cover those really quickly. Okay, so we can think of plant defense in terms of resistance traits. So resistance is the ability to avoid or reduce the amount of damage. So perhaps a plant is investing in chemical weapons, right, to keep the insects from eating them. Perhaps the plant is developing, has trichomes, and that keeps the insects from eating the plant. Or perhaps we're developing biochemical forms of resistance, like resistance to herbicides. Okay, so basically, if a plant has a resistance mechanism, it's able to avoid or reduce the amount of damage that it can get. Another important concept in plant defense is tolerance. And so this is the ability to buffer the negative fitness effects of damage. And the way that we measure this is we grow plants in the presence of the damaging agent and the absence of the damaging agent, and we effectively look at how fitness changes or doesn't. We think of tolerance, so basically a perfectly tolerant plant would have no change in the production of seeds in the presence of damage compared to the control, okay? We think of tolerance as plants that are able to have rapid regrowth after damage, or possibly they're releasing dormant apical meristems. Another way to deal with damage as a plant is to escape. So in escape traits, a plant avoids a stress by changing the timing of life history events. So perhaps they're producing seeds that can go dormant and can just delay germination. So they're staying away from whatever selective event is going to happen. Perhaps they can grow really fast to flowering, and they can flower early. Okay, so maybe you flower early before the damaging agent comes in. That's also a form of escape. Okay, so we've got resistance, tolerance, escape. And weeds also show really high growth, aggressive growth, and plasticity. So the ability to change when exposed to particular environments. Okay, so these are traits of weeds that allow them to persist in different environments. Okay, so here's the part of the talk where I want to get kind of cringy, right? So I've been studying plant defense for quite a while, and I've been thinking about these terms. I've been thinking about resistance. I've been thinking about tolerance, escape, and plasticity. And I want to think about these forms of defense and strategies in terms of strategies that we can apply to our own work when life gets stressful for, say, reasons such as this, where we're losing access to federal funds for various reasons that we can't control. What we can control is how we respond to that, right? And so what I want us to do in a very cringy way is to think of it like Bertha Wegman. What sort of characteristics of these persistent plant species that do all of these different strategies to just survive and persist? What can we take from those weeds to then survive and persist in a time in which we have a group of people that don't want us to be doing the science that we want to do, okay? So I like to think of it, what lessons can we learn from weeds? Well, there's resistance, right? We can always write our Congress people. We can always, show up and do active forms of, we're trying to promote funding for science, talk to people, all of those sorts of things. We can tolerate it, right? We can be under threat and we can be stressed out by not having access to research funds, and we can still be productive, right? So we can deal with a little bit of mental damage trying to do our science in an environment where it's hostile to science, right? We can escape it, right? We can avoid the stress. We can take a break. We can say, like, I don't have to think about what's happening federally in terms of funding rates. We can also, if we see an advantage or a time in which we can actually take advantage of something and have aggressive growth in our science or our work, we should take it, right? Okay. So what sort of lessons can we take from weeds to then be able to go on and be productive scientists in the face of almost hostile, difficult odds, right? So that's what I hope to kind of leave you with. I do have one other thing to think about. So this is kind of what we can control, what we can mitigate on our own as single humans in this world. There's also the component of our community that we need to think about, right? And I am the incoming president of the Society for the Study of Evolution. And as part of this, I started doing surveys over the summer to ask current faculty, assistant professors, associate full professors, a couple postdocs, a couple grad students about their feelings about the federal funding landscape right now. And so I want to share that with you because I'm thinking their responses and thoughts might stimulate ideas about how we can work as a community together. Okay. Oh, right. Plasticity. I forgot plasticity. Okay. So I asked people through surveys, how are you feeling about the future? Where 0 was very worried, 5 was not worried at all. So I did these interviews between May and July of this summer. And we can see that the full profs, they're still worried, right? But they're less worried than the assistant and associate professors. Then I asked, rate your concern about threats to academia. And here the scale was the same. I've got the numbers above the bars are the number of individuals that contributed. And we can see that assistant and associate professors are very concerned about academia right now in the federal funding landscape. Full professors are as well, right? So we're around 1 with those responses. And then I asked them, can you help me think of ways that the Society for the Study of Evolution can best advocate for its members? And so we came up with some ideas. The numbers up there for each of these boxes are the number of times this theme reoccurred across interviews. And so effectively people were asking, well, you can do advocacy for science funding and policy engagement, organize letter writing campaigns, advocate for continued or increased federal funding, train members in science advocacy and visit Congress, push back against funding cuts collectively or with other societies. The next thing was collaborate with other societies. So join forces to amplify your voice, coordinate actions with like-minded societies, and work together to defend science and respond to threats. A really big theme throughout all of my questions was people want support for early career researchers. So they said, provide small or travel grants for grad students and postdocs, offer mentorship and networking opportunities, prioritize support for junior scientists, including pre-tenure faculty. In the realm of tenure, the ideas were help advocate for tenure of evolutionary biologists just in general to keep them in jobs, provide letters of support and educational materials for administrators and tenure committees about the current federal funding landscape. They also said, start thinking about public communication and outreach, help members communicate to the broader societal impact of their work, educate the public, highlight contributions of evolutionary biology outside of academia. And then do some community building and member communication. So foster community support and morale, provide consistent communication on SSE's values, facilitate intra-member communication and cohesion. Then I asked them, and this is my last slide, I asked them, what are your top priorities to ensure the stability of the field? A lot of these answers were the same as the previous question. People want support for early career researchers, so try to provide little grants for postdocs and grad students, advocate for more science funding, do community and institutional support. Some new ideas that came up during this conversation was to think through as a society how to do more affordable and scalable science. So promote the use of open data, collaborative tools, and low-cost research methods, provide training on doing science with limited resources, recognize and reward research using published or teaching-centered data. So kind of open our ideas about what it means to use data that's already available rather than data generation. People also wanted us to focus on equity, diversity, and inclusion, so maintain and expand diverse recruitment at all training levels, protect and include international and marginalized scientists, ensure continued support for those most at risk in unstable funding climates. And finally, one of the ideas I wasn't expecting was to help develop education and training infrastructure. So create new or support existing training platforms. So the idea was basically like, what if we have fewer people that can come into the field of evolutionary biology and learn the basic science, learn the lessons? Well, perhaps as senior faculty, we can develop courses that we record and put somewhere for people to pull back out and use later. So kind of capture the knowledge that we have now, which is, I think, a great idea, but it also really bums me out to think about that. Okay, improve data sharing and training for non-traditional scientific outputs and equip scientists with quantitative and transferable skills for broader employability. So that was focus more on stats, command line code training for jobs in industry. Okay, so I hope those ideas have helped spark ideas for you that you could potentially think about both personally, how can we deal with the stressful funding climate right now, as well as work within your community and develop new ideas for support for one another. So if you have any questions, that's my last slide. So thanks. Yeah.
Yeah, curious whether you either looked at or thought about looking at how weeds act like in their non-native habitat compared to maybe their native habitat, or if it's like a native weed be like very urban to like wild setting, or maybe that ship has sailed because of settings.
Yeah. No, that's a great question. And I just wrote a proposal to not compare weeds in urban situations to their native habitat, but to compare the same weed species in agricultural landscapes to urban landscapes to suburban landscapes. So I would love to get it sort of the native, but I don't know if like the ancestral native environment in which the weed evolved is still present, right? But yeah, no, I like that idea. Yes.
So I was thinking about within that great slide you had of the different mechanisms of avoiding heat, resisting, tolerating, escaping. And then within the vigorous growth part, is there any idea about weeds overcompensating more compared to non-weedy species? Like within that, is there an idea that maybe they actually would benefit more than other?
Yeah, I mean, that's a great point. I had overcompensation in my talk yesterday. I didn't even think to put it in this talk. So thank you for that. But that's a great idea. I should have had overcompensation there. Yeah, that would be a nice, I mean, I don't know, like how would, folding back into the cringe just fully, like how would we incorporate the concept of overcompensation in our own lives? Like, because I don't want to say overwork, right? That's overcompensation to me sounds like overwork, but
I want to bring that back. Like as a form of trade, so our weeds more likely to overcompensate the scientific realm.
Yeah. Cool. Anything else? Nope. Thank you for listening to almost an hour about weeds. Appreciate it. Thanks. Okay. Okay. Thank you.
So your student Grace,